I'm Yaning Qiu. I'm an assistant professor from Wageningen University and Research. And today I will talk about the language model assisted data extraction and QAQC for microplastic health risk assessment. Mm, Before we really going into microplastics, so first let's see some questions. Every day we ask this kind of questions. So how long it will take to get to the zoo? How will the weather be tomorrow? Or probably one of the most timeliness question is which teams will advance from the group stage of the World Cup? And at the same time, there is a lot of data generated by multiple activities, from our clicking on phones, from our navigation data, or from all kinds of sensors, lab experiments, surveys, and also documents and messages. All the data can be useful to answer the questions if they are treated properly. And today my talk will be about how to use AI to facilitate the data treatment process, which is also the bridge between the questions and the data. So now we have a question, how to assess the risk of the microplastics to human health. And then to answer this big, broad question, then we need to divide it to several sub-questions. So for instance, how much microplastics are we exposed to? How much do we take in? How much remains in the body? And what are the effects of what remains? And to answer these questions in a meaningful way, then we need data. We need data to be accessible, reliable, and comparable. And this is also where we face major challenges. The data are exploding and they are scattered and unstructured across many publications, collected with different methods and reported in an inconsistent way. So therefore, we need some methods to improve the situation. And I will talk about how we tackle this through multiple methods. As Hannah has mentioned before, so microplastics research expanding rapidly. And there are more than 40,000 papers published during the past a bit more than 2 decades. And a recent perspective paper also argues that the growing volume of publications now exceeds the community's capacity to track, synthesize, and translate new information into actionable knowledge. And this also highlights the need to better systematically to filter, structure, and mobilize the evidence. And please remember this paper, Lost in a Sea of Information, the Microplastic Publication Deluge, because later I will go back to it. However, the increasing number of publications is not only a problem in microplastic domain. So many scientific fields are facing the same challenge. We produce more and more papers and text data, but our capacity to manually read, extract, and synthesize evidence is limited. And that's why many methods have been developed over the years. So over the past decades, the extraction methods can evolved from manual extraction to rule-based matching, machine learning to transformers and language models. And the semantic representations or the embedding models that Hannah just mentioned in her presentation is a bridge to bridge all the advanced methods. So here I also want to emphasize that these methods are not mutually exclusive. In practice, they can also work together, just like the embeddings work through different stages and involved during the whole improvement of the methods. So rules can provide structures, and also the language models can help generate structured outputs under a predefined schema. So we developed a workflow to adopt every of the advantages of each method and try to fit for our purpose. So how does our workflow work like? So we start with individual source papers, and these papers are retrieved automatically through licensed publishers' APIs. Then for each paper, we provide the language model some extraction prompt and also a structured JSON schema, which defines how the data should look like in the outputs. And then nowadays, most of the language models, the OpenAI or the Gemini models support this kind of structure outputs. So this means we can ask the model not only to understand the paper and extract relevant information, but also to return the results directly in a predefined JSON format, which, for instance, there can be different names for the country, like United States of America and USA and United States and America. And with JSON schema, we can unify them automatically just before we get the results, just by the language models. So here, the paper that I showed is an example of the extracted output, which is also a structured and like a structured format. And the paper here I show is a quite extensive paper because it covers more than 1,000 fish species from more than 1,000 fish samples from 37 species. And from this single paper, this workflow extracted 75 records, and for each of the records, it contains 39 different fields. For instance, polymer type, polymer shape, the location, the coordinates, and the tissue of the fish or the fish species. So you can imagine there are more than 3,000 field level decisions to make this whole extraction complete. And it is almost not doable by a human, like a human annotator. But sometimes we also know that language models are not perfect. They have their own issues. For instance, one of the most commonly known one is a hallucination. Therefore, in our workflow, we also try to adopt several validation and post-processing process to improve the reliability of the results. So for instance, for the extraction process, to improve the recall of the model, we run the models repeatedly for several times so that the model can fill the missing fields in a single run. And we also run a majority vote to make sure that different runs have a consensus on the data extracted. Once we get the completed data, we also use the cross-model validation to compare the outputs from different models, and therefore we can identify the uncertain records and keep the consistent ones. And to evaluate the extraction performance, we also would compare the AI extracted results with human extracted data. We do have a small human extracted sample for comparison, but one important point here is that we found like human extraction is usually not the most exhaustive at the most detailed level. In our case, human extraction results are usually relatively high-level summary values, such as paper level or category level main concentrations. And they usually do not extract every possible record and every field level decision from each paper. So to make them comparable, to make the language model's most detailed extraction comparable to the human relatively high level extraction, then we compare the paper level main values from human extraction and AI extractions here. And these three plots show this comparison results. Each point represents a paper level main value, and then overall most points fall close to the one-to-one line. So which suggests that the language model's extracted values are highly consistent with the human extracted values after validation. And this is the first part of the data treatment. So first, to get the data accessible. The next step to get the data reliable. So additionally, for risk assessment, the data, many microplastics also have different experimental design, contamination control, and reporting quality. Therefore, we need some kind of systematic way to assess them and to know which ones are more useful for risk assessment. Our previous research used a manual QA/QC approach and analyzed 45 papers with human assessments for 9 different criteria. And in last year, we published another paper using the language models ones. And this one, we can adopt this one to much more paper volumes than the manual version. More importantly, we found that the language model-based assessments is highly consistent with expert scores. So this means that the AI can meaningfully support the QA/QC approach and to support the data reliability assessment. The last part of the data requirement is to make them comparable. So for instance, in these two samples, they have different size, the size of measurements. And you would expect that in sample A, actually there are a lot of particles missing, like this, because they have a bigger size detection limit. And therefore, these two samples are not directly comparable. And this applies to almost every research. So to make them comparable, some kind of rescaling method should be used, like this. Here is not like I use a lot of AI tools to help with my coding, but I think the rescaling method is mostly statistics, so it falls beyond the scope of this presentation, so I will just skip it. Then with all the methods, now we can solve the problems to get the accessible, reliable, and comparable data. And probably it is possible to answer some part of these questions and to help assess the risk of microplastic to human health. I will just show some examples because the time is limited. So first, how much we are exposed to, and the second one is the effects. Here, so for instance, we extracted around 5,000 records from all the information that we could, all the papers we could get on the air we breathe, the water we drink, and all kinds of different foods we eat. After all the strict and self-verification and cross-validation, then we can get a global map of, for instance, here is the air concentration. And they were measured and reported heterogeneously, but they can be useful to identify global scale patterns if these scattered data are retrieved and processed properly. Same for the drink data. And probably you can see in the Mediterranean Sea, there is a yellow point, which means it has a relatively high value. Actually, it also agrees with the NOAA's marine microplastic records. So I think it's really interesting to see that the data from different publications actually aligns with each other. And here is a food one. We can, of course, do something different to the data because we have so many different fields. So for instance, here is a food polymer diversity, which can be calculated across globally. And for the effects, it's still at a very preliminary stage. So I'm not very familiar with that. So I'm still exploring what can be extracted and what should be extracted for risk assessment. So for instance, for now, I know a little bit that the polymer types that used in the effect tests are mostly polystyrene. And most of the ingestion routes are oral and dietary ingestion routes. Then there are different endpoint themes, so some are more common and some are less common. Whereas there are also some co-exposures, like the organic pollutants and metals are usually the most common ones for co-exposure experiments with microplastics. And now with some take-home messages. So first, AI-assisted extraction can transform literature into structured and reusable evidence. And the heterogeneous data can reveal patterns and help answer risk assessment questions. And the last one, just to reflect to the beginning, the prospective paper, AI can act as a drainage system for the microplastics publication deluge. So thank you. Thank you for listening, and I'm happy to take any questions.